Hello everyone, this is Sai Kumar. Today I am going to deal with photosynthesis, which is the basis of life on earth. So let us discuss about photosynthesis in detail. So coming to the introductory part, you may have some questions like why only the plant synthesizes the food and animals cannot do it. The major thing is plant contains a green pigment called chlorophyll, which is helpful in the trapping of light energy from the sunlight and convert it into chemical energy. So plants are called as autotrophs and animals are called as heterotrophs because plant can synthesize their own food, right? And whereas the animals cannot synthesize their own food and they dependent on the plants for their food purposes. So that is why plants are autotrophs and animals are heterotrophs. So why it is important to study the photosynthesis? The thing is, photosynthesis is the primary source of food on the earth, and secondary part is oxygen is liberated during photosynthesis, which is a very precious gas on this earth for our survival. So that is why we need to study the photosynthesis. And the thing is, you can define a photosynthesis in a way that it is an anabolic process where the simpler molecules like carbon dioxide and water utilized to produce the glucose in the presence of light. You can see in the reaction that 6 molecules of carbon dioxide are combining with 6 molecules of water in the presence of sunlight, and using the chlorophyll molecule, glucose is synthesized and oxygen is liberated as a byproduct. That is what the photosynthesis is. It is basically an anabolic process. Okay. Next one is, you may have studied some of the experiments conducted by various scientists in your previous classes. You are very familiar with this bell jar experiment, which is conducted by Joseph Priestley, and hydrilla experiment, which was conducted by John Ingenhousz. These two experiments, they concluded that oxygen is liberated during photosynthesis. Next, T.W. Engelmann, he is another scientist who used a different approach to study the photosynthesis, where he used a green algae called cladophora, which are suspended in the aerobic bacterium. Aerobic bacteria in the sense they can survive only in the presence of oxygen. Here Engelmann used the prism to split the light into 7 colors, that is VIBGYOR, where the blue light and red light regions shown that oxygen is liberated more. That is why he concluded that because the aerobic bacteria are formed in the clusters at these regions, red as well as blue regions. You can see that clearly in the image. So that is why the photosynthetic rates are high in the blue regions as well as red regions, where the green rate is reflected by the plants or the algae. So that is why the rate of photosynthesis is very low in the green light. This is what the T.W. Engelmann's experiment. Coming to the next part, electromagnetic spectrum. The only visible part is of light is around 380 nanometers to 750 nanometers. That is what the electromagnetic spectrum is, and this visible light is only useful for the plant to photosynthesize. Next, the absorption spectrum as well as action spectrum. Absorption spectrum and action spectrum are the graphical representations of the photosynthetic absorption and rates of the photosynthesis. So it is generally a graphical representation. You can see in the above diagram, absorption spectrum is a graph which represents the absorption of different wavelength of light by the photosynthetic pigments. You can see the photosynthetic pigments like chlorophyll, xanthophylls and chlorophyll B, they are absorbing light at different wavelengths. The more absorption is seen in the blue light and red regions. Next one is action spectrum. It represents the rate of photosynthesis at different wavelengths of light. As already proven by Engelmann's experiment, the rate of photosynthesis is much higher in the blue region as well as red region, and the green region has the least photosynthetic activity. This is what the absorption and action spectrum is. Coming to the next part, where the photosynthesis takes place. You may have been wondered that all the parts of the plant are looking green when they are young, and when they are matured, the leaves are only young in color, green in color, where the photosynthesis takes place. So the green colored parts of the plants, they perform the photosynthesis. So why? Because the leaves of the plants contain the specialized cell organelle called as chloroplast, where they contain the chlorophyll pigment which carry out the photosynthesis. The chloroplast is a membrane structure which consists of stroma, granum and stroma lamellae. Granum consists of the group of thylakoids which are coin-like structures arranged one upon another like a stalk, where they contain the chlorophyll pigments. And the stroma contains the fluidic content where there will be taking the enzymatic reactions. And stroma lamella is the connection between two or three or multiple granums. Okay, so what is light reaction and what is dark reaction? Light reactions are the chemical reactions which takes place only in the presence of light, and dark reactions are the reactions which are independent of light. Here the light reaction products are ATP, NADPH and oxygen, whereas the dark reactions products are the glucose. So let us discuss what is the light reaction and what is dark reaction. Light reaction is called as a photochemical reaction and the dark reactions are called as carbon reactions. So let us see the block diagram of the photosynthesis in the next slide. So let us see what is a pigment. A pigment is a molecule which has the ability to absorb different wavelengths of light. These photosynthetic pigments in the leaf can be separated by a technique called chromatography. The various pigments in the plants are chlorophyll A, chlorophyll B, xanthophyll and carotenes. Chlorophylls are the molecules which contain a structure called porphyrin head and a phytol tail. You can see in the diagram. The porphyrin head consists of the central atom called magnesium and the phytol tail is a chain of hydrocarbons. So you can see the molecular formulas of those pigments clearly and their colors. So blue green color of chlorophyll A, yellow green color of chlorophyll B, xanthophylls contain the yellow color and carotenes contain yellow to orange color. So chlorophyll B and xanthophylls and carotenes, they act as the accessory pigments. So here the chlorophyll A is present at the center and they are surrounded by these accessory pigments, and these pigments they are combined with the proteins which is called as a photosystem, right, which is called as a photosystem, where the chlorophyll A is the central molecule and remaining chlorophyll B, xanthophylls and carotenes, they surrounds the chlorophyll A. The chlorophyll A acts as the reaction center. So these carotenes especially they protect the chlorophyll A from photooxidation. So a photosystem is nothing but the aggregation of the pigment molecules along with some proteins. They forms a complex, that is a photosystem, where the reactions takes place. So next one is a light reaction and dark reaction. Light reaction is a reaction which takes place only in the presence of light, where the photolysis takes place. So what is photolysis? Photolysis is the splitting of water molecule into protons and a molecule of oxygen or an atom of oxygen. Here the oxygen is liberated in the light reaction and ATP and NADPH are synthesized in the light reaction. So these ATP and NADPH synthesized in the light reaction, they are utilized by the dark reaction cycles or the reactions. Here in the dark reaction, the CO2 from the atmosphere is absorbed by the leaf and they undergo a cycle called Calvin cycle, where the carbon dioxide is reduced to produce the carbohydrates. Here the ATP and NADPH are utilized. So that is what the light and dark reaction is. And during the light reactions, so how these ATP and NADPH are synthesized? When the light falls on the thylakoid membranes, they contain the photosystems. So these light excites the electrons present in the pigment molecules, and these electrons are passed through photosystem 1, photosystem 2 through several proteins called electron transport chain, where the potential gradient generates the ATP. So that is how the ATP and NADPH are formed in the light reactions. So this is what about photosynthesis in brief. Thank you for your attention. Thank you.